Soy Juan Alberto Alcántara y a nombre del Instituto Politécnico Nacional y del CIEMAD, en el marco de los festejos del 84 aniversario del IPN, es un gusto darle la más cordial bienvenida a esta sesión donde escucharemos la conferencia Micronanoplastics in Human and Animal System. Micronanoplásticos en sistemas humanos y animales. Actividad que forma parte del programa de la Cátedra Patrimonial en Medio Ambiente, Héctor Mayagoitia Domínguez, en su versión 2020, en su quinta edición. Presento a nuestro ponente del día, que nos hace el favor de acompañarnos. Me da mucho gusto darle la bienvenida al director del Centro de Nanobiotecnología del Instituto de Tecnología de Vellore, al doctor Chandrasekaran. Sea usted bienvenido y muchas gracias por aceptar la invitación. You are welcome and thank you very much for accepting the invitation.
Thank you.
La dinámica de la sesión será la siguiente. El ponente tendrá 45 minutos para su exposición y posteriormente tendremos una sesión de preguntas y respuestas de 15 minutos. The dynamics of the session will be follows. The speaker will have 45 minutes for his presentation. Later, will we have a question and answer session of 50 minutes. Les recordamos que pueden hacer las preguntas a todos nuestros, las personas que nos siguen por el canal de YouTube, indicando su nombre, la institución a que pertenece. Y bueno, pues aprovecho también para solicitarles que se suscriban a nuestro canal de YouTube. Antes de pasarle el micrófono a nuestro ponente, me permito compartirles un resumen de su semblanza curricular. El profesor Chandrasekaran, director del Centro de Nanobiotecnología del Instituto de Tecnología de Vellore, en la India, ha realizado varias contribuciones conceptualmente importantes y ampliamente citadas al área de la nanobiotecnología. Ha realizado contribuciones pioneras en la preparación y aplicación de perlas de biopolímero de nanoemulsión de NEM para el control de infecciones bacterianas en animales de incubación bajo el desarrollo de la tecnología DST, películas de biopolímero impregnadas de nanoemulsión para el envasado de frutas para mejorar la vida útil de estas, ¿no? El doctor tiene más de 12 patentes presentadas, publicadas en el área de nanobiotecnología, 278 publicaciones en nanobiociencia, y su total de citas son 9,428 citas. Ha completado 22 proyectos financiados. Es un revisor destacado en revistas de renombre. Es miembro de la revista Royal Society for Biology y miembro de la Royal Entomological Society of London y miembro de la Royal Society of Chemistry. Además, que ha sido mentor de 19 doctorantes. También, aprovechando, le doy una cordial bienvenida al doctor Jonathan, profesor del CIEMAD, para apoyarnos en la traducción de las preguntas y respuestas. Dr. Chandrasekaran,
yeah.
We hear you. Please.
Thank you. Okay, my screen is visible?
Yes.
Okay, can I proceed?
Yes. Come.
Okay, thank you. Very nice meeting everybody on this online platform to deliver my lecture for Dr. Héctor Mayagoitia lecture series, which is being organized by the National Polytechnic Institute, Mexico City. So I thank Dr. Jonathan and his team to organize this wonderful lecture. And I'm going to deliver my talk on micro nanoplastics in human and animal system. So I am Professor Chandrasekaran from Vellore Institute of Technology, India. This is also known as VIT in India. So VIT India, which is which got IOE status very recently, Institute of Eminence by our Government of India. So I'm going to the next slide. So this will be my lecture topic for the day. So I will cover, start with the introduction, then what are the plastics, what are its uses, and plastics and human health, is there anything like that? Then micro nanoplastics, then assessing the risk, then conclusion. So in the micro nanoplastics, how micro nanoplastics enter the environment and who is making micro nanoplastics? Is there any system that is making it? And assessing the risk, what is the leaching of plastic and what are the components that is there in the plastics that could leach into the environmental system? To have a brief introduction on micro and nanoplastics, normally the megaplastics, the plastics that we use in our day-to-day life, is undergo weathering process. So when we are using plastics, the plastics normally, any material in this world will wither. So plastics normally wither in the environment and different particle size come into being in the environment due to weathering process. So right from megaplastics, there are a list of plastics, macroplastics, mesoplastics, microplastics, and nanoplastics. So only these two, micro and nanoplastics, are very much researched and also very much experimented in our recent times, because microplastics ranges from 1 to 5,000 micrometer, and nanoplastics, that is from 0.1 micrometer or less than 0.1 micrometer. Because these two plastics are used in most of the consumer products, especially nanoplastics are used in a lot of cosmetics, and also microplastics in face cleansers and other cosmetic items. So these two are very, very dangerous when it is dumped in the environment. Because of natural weathering process, they undergo many changes and ultimately hamper or give a destructive result to the environmental system. So these are the types of microplastics. One is a fiber, that is from our cloth material. And these are all the pellets, the fragments, the films, the foam, and microbeads. So these are all the types of microplastics that is there in the environment, that is all coming from the consumer products, and it's all man-made. So plastic invention was something around 50s or late 50s, early 50s and 50s. The synthetic polymer came into being. During such time, it was very useful because plastic are very useful in our day-to-day life. So it is very difficult to replace plastics. So now people started to understand the various ill effect of plastic to the environmental system. This is one of our survey which we did in Chennai coastal region. So we did a big survey in the coastal area of Chennai, and this is Kasimedu. This is the place where there's a fisherman folk lives. It's a fisherman coastal region. And we could able to see many different kinds of plastic in this area, many different types. And among these, the most predominant plastic that we could able to identify was polystyrene and fibers, polyethylene fibers. So this is me. I'm doing this survey. I'm interviewing a fisherman. And this we did in collaboration with our School of Social Science and Languages, VIT. So we could able to find out two dominant plastic material. One is polystyrene. The other one is the fiber, the plastic fibers that comes from the fishermen ropes and polystyrene plates and cups. It is thrown by tourists. They visit this place. There's some places here or tourists. They commonly visit this place. So along when they come, they carry food. This is a takeaway food, and the takeaway foods are packed in polystyrene packets, and they simply throw onto the sea, onto the shore, and they just leave. So this was very pathetic when we saw this place. And the other place where we did a survey in southern tip of India, that is along with the Fisheries Management Division, that's called the Indian Council for Agricultural Research. We did a marine survey, the Marine Fisheries Institute that is there in Ramanad, that's Ramanathapuram, that is in Tamil Nadu. We could able to see 22 different plastic samples. So we collected from Indian Ocean within 100 square meter area of sea. So every 100 meter, we could able to identify 22 different plastic samples. See, these are the plastic samples. And most of the plastic samples, polyethylene types, most of these. And these are all fibers, the ropes they used by the fishermen folk. And these samples we characterized and we used for various toxicological studies. So this is the plastic pollution scenario in the world, that more than 8.3 billion tons of plastic has been produced since early 1950s, out of which 60% of the plastic has ended up in either a landfill or natural ecosystem. So even people talk about plastics in agricultural ecosystem. Why? Because most of the surface runoff, most of the sewage, most of the drains, they end up in sewage treatment plants. In the sewage treatment plant, there are no technology to remove microplastics or nanoplastics. So all ends up in sewage sludge. Then ultimately, these sewage sludge are used in the agriculture field as an alternative to fertilizer. So in turn, most of these microplastics, they enter the agricultural food chain. So this is another area that needs to be concentrated in future. So these are micro nanoplastics. So that means depending upon the size, we categorize this as a micro or nano. So they break, the megaplastic breakdown into micro nanoplastic due to weathering process. And they range in size from that of a grain of rice all the way down to a virus. So this is a complete weathering process of it. Now, scientists have proven that the nanoplastic that is present in the sea could enhance the ocean acidification. There are some group working in UK, they have found out that it can add up to ocean acidification process, these nanoplastics. When they enter the planktonic form, they enhance acidification process. So these are some of the recent findings that scientists have come out with. So these are the different categorization of plastics: the macroplastic, mesoplastics, microplastics, and micro and nanoplastic. This is a nanoplastic. This is a TEM image of a nanoplastic, which is less than 1 micrometer in size. Look like a colloids. It's almost like a colloid. So in our laboratory, we did some work on how to isolate plastic. So we fabricated some instrument using electromagnetic system, and we could able to isolate microplastics from different matrices, from different environmental matrices. So this is a setup that we got a published patent from India, and we could able to extract different types of different microplastics from different matrices. I'll show you. So this is the isolated micro and nanoplastics. This is from microplastic from sea salt, microplastics from marine water, and this is from microplastics from lake water, and this is nanoplastics from face cream. We could able to identify the isolate, and microplastics from treatment plant, the sewage treatment plant, and microplastics from effluent waters, and these are nanoplastics from shampoos. We could able to identify nanoplastics from shampoos, and this is a body lotion. So where all this plastic ends up, it ends up in the sewage treatment plant ultimately. And from sewage treatment plant, it goes into the sludge, sewage sludge. Then ultimately, it goes to the agricultural ecosystem. The remaining part will get washed off from the drains into the aquatic system and to the marine system. So unless until we control this process, the usage of plastic to some extent, it will be very difficult. Because I still remember when I was in Germany, this usage of plastic in the year 1999 and 2000, they started to curtail usage of plastics. So if anybody visits a convenience store, they won't give plastics free of cost, plastic bags. But now in India, it is very difficult to get plastic bags free of cost, because we have to pay money and get the bag. So now most of the shops in India, they have abandoned this usage of single-use plastics. The problem comes when we use a plastic for one-time purpose. So these plastics having now being proven to cause environmental effect as well as the human effect. So what we did a small study in a lake water ecosystem, and also we compared with the seawater system. So this was a small microcosm study where we took a polystyrene nanoparticle. It's a polystyrene, virgin polystyrene, and we could be able to see over a period of time, the size of the particles started to increase. So there could be two possibilities. One is the colloids in the seawater and the lake water would add up to the particle size, or it is the aggregation of these nanopolystyrene, they aggregate together and become a bigger particle size. So when polystyrene, supposing if it combined with the colloidal system, the natural colloids in the water matrix, then there is 100% possibility that this could be engulfed by the living organism, because the living organism will sense it as a real system, sense it as a real food, and it will get, the particle will get into the system. So there are a lot of proof that we have also done the same experiment with Philodina rosella, which is a freshwater rotifer, and we have proven that this rotifer lifespan was shortened, and the rotifer could not jump to F2 generation. So it stops at F1 generation. So these are some of the findings we did in the lab, and trying to prove that micro nanoplastics are having greater impact to animal system. So once again, sedimentation of polystyrene nanoparticles in seawater. So we did an experiment with the seawater. The previous slide was on lake water. This was in seawater. After 4 hours, gradual increase in particle size of polystyrene nanoparticles was observed. That is from 2.5, 5.0, 7.5, and 10 centimeter depth. So we could be able to see along the depth, there was increase in the particle size of the nanopolystyrene. That means the colloid, the natural colloids are more in the bottom layer compared to the top layer of the system. So micro nanoplastics, occurrence in human health, ingestion of micro nanoplastics and uptake across the gut, how it enters the gut of the human, the interaction of microspheres and nanoparticles with cells and tissues, and interactions with biological materials and toxicity. Actually, if we look back just 2 decades, so those days people are working with heavy metal toxicity. Then heavy metal was during 1970, there was a program called muscle World Muscle Watch program. During such time, people started to work on heavy metal toxicity, heavy metal impact to the environmental system. Then people started to work on heavy metal speciation. So how different species of metal differs in terms of its toxicity. Then slowly, now we started with nanomaterial and nanoparticle toxicity. Then after that, it is ionic toxicity from the nanoparticles. Now, the recent field of research is micro and nanoplastic toxicity. So this is also a sort of nanomaterial, nanoplastics. So it's a development in toxicological research, right from heavy metal to the micro nanoplastics toxicity. This is a simple flow chart just to have a glimpse on how these micro and nanoplastics are created or generated in the environment. So from this, PVC is a polyvinyl chloride, polyethylene, polystyrene. Along with that, there are additives, phthalate, bisphenol, heavy metals, so many additives are there. These are all known as plastics. And all the plastic material, they get weathered. Weathering in the process, UV, heat, temperature change, so on and so forth. And microplastics from consumer, face cream, cleanser, toothpaste, everywhere. That also get weathered. So this weathering coming to a microplastic, then microplastic weathering to nanoplastics. These are priority pollutants in the environment, that is POP and heavy metals. They also act as a vector. That means these nanoplastics and microplastic, they carry these priority pollutant to other non-source pollution points. So these are the additives. Normally they add along with plastics when they are getting manufactured. Micro nanoplastics and human health. So majority of studies have documented microplastics in the guts of organisms. That means an organism that is not generally consumed directly by human. And exceptions to this include shellfish such as mussels and clams that are eaten as whole. And ingestion of micro nanoplastics by human directly is through consumption of dry fish. This is another big area, dry fish. Some dry fish, they don't, because dry fish, smaller fishes, they don't clean the intestine, they simply dry it. So these fishes will have nanoplastics or microplastics, so they consume as a whole. So these are some of the route that microplastics, nanoplastic, directly or indirectly influence human system. So this is a small information on how it enters the intestine, the ingestion of micro nanoplastics and uptake across the gut. A diagram illustrating a proposed recirculation pathway of polymer nanoparticles. So after oral administration, the nanoparticles are taken up into the bloodstream from the gut through M cells. M cells are nothing but the epithelial cells. From there, through the lymphatic system and into the liver and gallbladder. The particles are then re-released into the gut together with bile. So it enters the bile before excretion in feces and urine. So this is the route of micro nanoplastics when it is ingested by a human through food directly or indirectly. So what happens? So interaction of microspheres and nanoparticles with cells and tissues. So normally food packing material and nanomedicine. That means we pack most of the foods packed in India, or most of the material are all single-use plastics. So they pack food. If you take any big restaurant, they pack, especially Indian foods are having almost 23 variety of side dishes during lunchtime. You can find they pack most of the foods in plastic bags. Now slowly it is getting abandoned actually, but still in some of the hotels and restaurants, they pack foods in single-use plastic bags. So what happened when they pack the food, there is always a possibility that the plastics get withered along with the food that is hot in it. So there is some sort of weathering, and there is a possibility that these particles would get imbibed into the food material. And certainly, in vivo behavior will be dependent on numerous factors such as physico-chemical properties of particle size. So there are so many other parameters that could influence the pathway of these micro nanoplastics. One is the size, the surface charge, the aspect ratio, porosity, and surface corona. Corona is nothing but the formation of the organic onto the micro nanoplastics. And risk assessment of manufactured nanomaterial. So the toxicity of titanium dioxide and carbon, they have shown it is comparable with that of nanopolymers. That means the nanomaterial toxicity, they are compared with that of nanopolymer in terms of uptake across the gut into the circulation and redistribution to liver and spleen. So the toxicity and risk assessment of manufactured materials, they are more similar, the nanomaterials similar to the nanopolymers. And circulation time is highly dependent on surface characteristics of the particle. So there is always a surface charges. Depending upon the surface charge and the size, the toxicity varies when it enters any system. So due to their surface properties, nanopolymers are predicted to absorb macromolecules. This is another big area actually. So I'll show you in my following slide. That's called the corona formation. That's called the protein corona formation. So when we interact protein, bovine serum albumin or human serum albumin, there is something called a corona that forms, a protein corona. That means the polystyrene or the nanoparticles or the nanopolystyrene, the microplastics, they get engulfed into the protein molecule and they form a corona. That may act as a self-material when it enters the human system. So these are some of the different types of plastics: polyethylene, polypropylene, polystyrene, polyethylene terephthalate, polyvinyl chloride. So depending upon the type of plastics, they have, accordingly, they have their additives in it. So these are some of the plastic, how humans are exposed to micro nanoplastics. One is through food packaging, food chain, cosmetics, household products, water, then air. Through air also they get this plastic material into the human system. So this is what we isolated from face cream, the microplastics. This is the different shaped microplastics that is isolated from face cream. So you see different sizes of these microplastics. They put these microplastics in face cream so that to have a friction, the face cleanser. So ultimately it is drained into the drainage system, ultimately reaches the aquatic and marine system. Now the effect. So how these micro nanoplastics, when it is interacted with human proteins, the human serum albumin and hemoglobin, what happens? So what we did, we took a virgin nanoplastics, which is 100 nanometer in scale. And this is the electron micrograph of polystyrene, virgin polystyrene. This is a polymer. When we interacted over a period of time, we could able to see protein corona formation of nanoplastics upon interaction with human serum albumin, HSA. So there is initially, after some time, there is a small circle around it. This is the protein molecule, and you see this is the nanoplastic. Over a period, after a period builds up, you could able to see the size of the corona increases slowly. That means it keeps on binding onto the. So what happens? Sometimes the cell will sense this as a self material. Why? Because of the corona formation that is happening due to the protein molecule. So when this system sends this foreign object as a self, then there are a lot of problems will come into picture. I'll show you one by one. So there is something called protein-induced coalescence of nanoparticles, the nanoplastics or the nanoparticles. See, from Figure C, if you see, two coronated nanoplastics by mean of non-specific protein-protein attraction. There is a thread-like structure. So after corona formation over a period of time, so they get coalescence, they get attracted to each other, and they slowly become bigger in shape. So this is the coalescence of particles. Slowly it is happening in this system. So we did the same experiment with a cell culture system. I'll show you those results. Here, D and E, corona reorientation. There's a neck-like bridge between two plastic nanoplastics, D and E. This is D and this is the E. So you could be able to see the corona reorientation, neck-like formation. Then F is the widening of the neck of bridge of diffusion of nanoplastics to coalesce with each other, and G and I is the coalesced nanoplastics inclined to a round shape with multi-layered human serum albumin corona. So, and G and H is the significant grain growth. So if you see, there is a significant growth of this corona layer forming onto the nanoplastics, so that the material will be sensed by the living cell as a self material, So it won't recognize that as a foreign body. If it recognizes as a foreign body, then there is a problem with the immune system. So it will recognize as a self material. This once again, this protein corona formation and nanoplastic coalescence effect on human blood plasma. That was human serum albumin. This is in the case of blood plasma, how this corona formation and neck formation and coalescence of these particles taking place. This is the coalescence effect, hard corona, soft corona, the grain-sized acceleration, and this is non-specific attraction, and this is the nanoplastics, and this is the coarsening of the nanoplastic with the protein molecule, and there's a neck formation. Then, after that, we could be able to see a protein conformational chain and further to denaturation of protein. So after the nanoplastics interact with the human serum albumin, we could be able to see there is primary and secondary structural changes happening in the protein molecule. So that we could be able to identify using a circular dichorism spectra and also the FTIR spectra of HSA of pre and post nanoplastics interaction. So we could be able to see a structural change and we could be able to confirm it. And this is a cytotoxic and hemolysis activity of coronated, virgin, isolated nanoplastics. Isolated means we isolated from face cream and other consumer products. So we could be able to see a cytotoxic effect compared to the control, and also a hemolysis also happening in the case of virgin and isolated nanoplastics. That means plasma coronated nanoplastic showed a significant toxicity on lymphocytes and erythrocytes due to the protein conformation changes. And also we did a genotoxic activity study using human lymphocytes, and we could be able to see a genotoxic effect and confirmed through Comet assay, and confirming that their DNA tail length was very high and observed cytotoxic and genotoxic effects that signifies that after interaction with nanoplastics, the plasma proteins turns into bio-incompatible and delivering an altered biological activity. That is also we could be able to see. So this is their range, and this is the plasma coronated nanoparticles and different concentration of virgin and the coronated nanoplastics. And also we isolated the nanoplastics from different consumer products. And we could be able to see that nanoplastics were able to induce genotoxic effect in human lymphocytes. Then we did a study with cells, the human Hackard cell line, that's a skin cell line. And we could be able to see using a fluorescent nanoplastics, we could be able to see that the cell were able to uptake nanoplastics only after 24 hours. That means what is happening before it? The cell was not able to recognize this as a self or non-self. That means Only after corona formation, only after the protein is bounded onto the nanoplastics, the cells were able to recognize that as a self material and able to engulf those material. Then what is happening after that? The corona formation on nanoplastics. This is what is happening. So slowly the corona started to build up after 28 to 40 hours, 48 hours. Then after 24 hours, these plastics, they got engulfed into the cell system. So nanoplastics are engulfed by cells, only multi-layered protein corona formation. So only after multi-layered protein formation, it was able to engulf. Because by the time the cell was not able to recognize that as a self material. And we could able to see a phagocytosis in the process. So there is something called phagocytosis, a phagolysosome we could able to see. This laser confocal microscopy, and the green fluorescent tagged nanoplastics, these are the green, and the red neutral dye is the neutral dye. The green mass fluorescent nanoparticles, nanoplastics in phagosomes. So these are the inside the phagosomes, and the yellow mass is a phagolysosome. So that means the cell is able to lyse these nanopolystyrene. Then what is the meaning of it is the polystyrene, which is a polymer, may be a possibility that it may lyse the polymer to a monomer. So we did an experiment and we could able to see some sort of denaturation of nanopolystyrene. That means what? The nanopolystyrene is being denatured by the cells. When the cells get engulfed, the phagocytosis is happening. Phagolysosomes come and does a phagocytosis, and it lyses due to the phagolysosomes. The nanoplastics are lysed and it is disintegrated and denatured. And we could able to see, or we could able to see a release of unit or a polymer unit, a subunit of nanopolystyrene, the styrene, in our GCMS analysis. That means what? Denaturation of nanopolystyrene was observed, which may lead to the release of monomers into the cell. The monomers here is styrene, and styrene means it is carcinogenic, which is cancer causing. So these are some of the experiments, the cell proliferation assay we could able to see. And we did an experiment with control, polystyrene, styrene, nanoplastics 1, nanoplastics 2 of different size ranges, and also the hydrogen peroxide. And we did with a Hackard cell line, a skin cell line, we could able to see a drastic fall in the cell viability when we did in different concentration after post-exposure time. And we could able to extract the nanopolystyrene extracted from female face scrubs, nanopolystyrene 2, has significant effect on cell proliferation index than nanopolystyrene 1 from male face scrub. So gradual decrease in the cell proliferation was observed from 24 hours post-phagocytosis. So this was the finding actually. And this paper, it is now in review in Autophagy, the journal. And this is the senescence. We could able to see the aging of the cell due to the nanopolystyrene. So senescence activity during post-phagocytosis. So after 72-hour post-phagocytosis, the cell started to die off. That means slowly it started to age. The cell aging we could be able to identify. And this is the A to C control at 24, 48, 72-hour post-phagocytosis. And D to F is the polystyrene nanoparticle at 24, 48, 72-hour post-phagocytosis. And G to I is the styrene. We took a monomer styrene at 24, 48, 72 hours. And J to L is the nanoplastics from male scrub at 24, 48, 72 hours. And M to O is the from the female face scrub. So we could able to see after post-phagocytosis. So we could able to see a senescence activity of the cell, the aging of the cell after post-phagocytosis due to nanopolystyrene or nanoplastics. And also at the same time, we could be able to see the microplastic effect in some of the model system, the plant system, and we could be able to find out that these micronanoplastics induce genotoxic effect in model plant system Allium cipar, the onion root tip cells. And these are the pictures of it, the chromosome aberration in the virgin nanoplastics, and also isolated nanoplastics from face frames. And this was the microplastic effect. We did an experiment with Artemia salina, and we collected these microplastics from the environment, and we fed the animal Artemia salina with these microplastics, and we found out that the hatchability of Artemia salina, the cyst, was delayed. There was a sharp decrease in their mortality. Increase in their mortality. That is 20 microgram per ml of microplastic sample inhibited 50% of Artemia salina cyst. So this was the finding. And mortality percentage of Artemia salina in presence of microplastics. So mortality rate, the Artemia salina in the presence of microplastics, 20 microgram per ml collected from this place. This is seashore belt in Ramanathapuram, Tamil Nadu. So these are the mortality rate for different types of plastic particle we collected from the environmental samples. And these are the effects of polystyrene nanoplastics on Artemia salina, the lethal indices, LC50 and LC90 in Artemia salina. Artemia is a zooplankton, which is a major feed for aquaculture fish in aquaculture. This effect of polystyrene nanoplastics on Artemia salina, we did the biochemical assay. We could be able to see the protein, how this protein of the Artemia salina varies after the introduction of these nanoplastics. And also, we could be able to see the reactive oxygen species and like lipid peroxidation. And lipid peroxidation activity evidences the easier penetration, membrane permeability of polystyrene spheres into Artemia salina tissues. And there was concentration dependent elevation was observed. And this is the effect of polystyrene nanoplastics on Artemia salina once again on the glutathione reductase assay and also the catalase activity. And we could be able to see the protein conformation changes and denaturation of protein when these microplastics, nanoplastics from the environment, they are interacted with the human serum albumin proteins. These are the interaction of polystyrene nanoplastics with biological macromolecule like amylase, bovine serum albumin, and this is the E. coli and Bacillus species. And this is the zebrafish RNA, and this is the purity of DNA, RNA, and post nanoplastics treatment. How the polystyrene interacted, the red one, and this is the control for different species of zebrafish tissues we have identified. The purity ratio we have done. And this is the cytotoxic and hemolysis activity of coronated, virgin, and isolated nanoplastics. So plasma coronated nanoplastics showed significant toxicity on lymphocytes and erythrocytes due to protein conformational changes. And this is the viability of polystyrene biomolecular complex treated cells. So how this PS protein complex and change in cell viability. And this is the different sugar molecule we have interacted with the nanopolystyrene. And we have introduced in the Hackett cell HCT116 cell viability study. And we have introduced and we have seen the viability of these cell lines. And this is the publication we got from one of our paper. This is our team working on micro nanoplastics. This also popped up in one of the famous newspaper, Hindustan Times in India. And this is another publication. This is the patent that we got published. These are the reference. Thank you very much for patience hearing. If there is any question, I'll be very happy to answer.
Gracias, doctor. Thanks, doctor.
Thank you very much. My pleasure.
Muy interesante presentación para el efecto que causa en los animales y en los humanos. Very interesting presentation for the effect caused in the animals and the humans. Ahora iniciaremos la sesión de preguntas y respuestas. Now we start the question and answer session. Por favor, Dr. Jonathan, le cedo la palabra. Please, Dr. Jonathan, I give you the floor. Hay una pregunta, la primera pregunta la voy a decir en español y el doctor la va a traducir. Hasta la fecha, en la comunidad científica y algunos más no aceptan que los microplásticos afecten la salud humana. ¿Me puede aclarar esto un poco más?
See, the question is from Dr. Sujita Suresh Babu. She's from IPN graduate. Till date, in scientific community, many don't accept that microplastics affect human health. How could we clarify, sir?
Yes. Yeah, very interesting question. That's what I told in my previous slide. The question still remains, because after the experiment that we have done with the human skin cell line using nanoplastics, we could be able to see styrene-like molecule releasing into the system after the coronation of this plastic happening. That means the protein, when it is wounded onto the nanoplastics, these plastics are disintegrated or degraded and broken down to monomer, and these monomers are toxic. That is already proven by Cancer Research Institute that styrene are highly carcinogenic. So there may be a possibility that it may cause cancer. This is one. The other thing is plastic as a virgin plastic may not be toxic in some cases, because when virgin plastics, when it adds up to some other thing, like it acts as a vector, it carry other heavy metals, other POPs, and other organic onto it, and it will enter this animal or human system. So plastic particle act as a vector for other pollutants. So if that is the case, that may cause an effect to the organism. That is one. The other thing is, doctor, the plastic, while, yes.
Can I stop the screen sharing, please?
Yes, please. Yeah.
Thanks. Continue,
so the other, yes. The other thing is the plastic during manufacturing, they add other material like plasticizer, the phthalate, the bisphenol, and along with that, other heavy metals. So there is a possibility that these leaching of these additives may cause toxic effect to the animal or human system. So these are the findings that people have already proven in animal, in aquatic animal, lower form animal, in bacterial system, and also in the human system. They are trying to prove with the different in vitro studies that micro nanoplastics impart a major impact to human health.
Thanks, doctor. Dr. Jonathan?
Thank you, doctor. And I will try to tell a short in Spanish. Plásticos, microplásticos o nanoplásticos, cuando entran en el sistema, hay una sinistrin, como molecular, es como monomers. Ese sí entra en el sistema después. Sí va a cambiar como nanoplásticos. Este está aprobado y también descubrieron en el Cancer Research Institute, Instituto de Cáncer, y también puede causar cáncer. Todavía están aprobando en diferentes maneras, en diferentes formas, cómo puede afectar directo. Hay un estudio de laboratorio también, y donde hacen estudios en plásticos vírgenes donde se agregan plastíceres. Esos plastíceres a veces puede provocar cáncer y también hay posibilidad por lixiviación de los metales en esos celulares. También hay estudios in vitro, todavía siguen en estos estudios. También si hay posibilidad por transportar o proceso de absorción o algo así para afectar salud humana. Todavía está en diferentes fases de estudios. Muchas gracias.
Thanks. The next question. Moisés Manzanares, en su mayoría los microplásticos son de plásticos reciclables o plásticos no reciclables. Pregunta.
Can you repeat the question?
Yes.
¿Puede repetir la pregunta?
Moisés Manzanares, en su mayoría los microplásticos son de plásticos reciclables o plásticos no reciclables, es la pregunta.
The major part of the plastics or nowadays recyclable plastics or biodegradable plastics. What is your opinion? Dr. Chandrasekaran?
Yes. Could you please repeat?
Nowadays, majority of the plastics are biodegradable or recyclable plastics. So, what is your opinion on this aspect? This question is from
that is right. The best example. Yeah, that's right. Yes, the recycling of plastic has taken place very recently. For example, in the agriculture system, people talk about mulch films. They put mulch films onto the system and they are replacing with biodegradable plastic. The other side is biodegradable plastic bags have come into being. But even that is a plastic, a polymer, any particle or any material will degrade in the environment, so naturally. So they will add some additive. The bioplastics will have some additive to have a plasticizer effect. If the change from the synthetic polymer to a natural polymer is not 100%, even now, you cannot say that 100% you can abolish plastic. Maybe you can change to a natural polymer to some extent, but it is very difficult to abolish total 100% the synthetic polymers. But we need to find a way out to how to manage without plastics. That is the best way, the best option. Or how to manage plastic waste. That's another best option. So here the point is, we should know how to manage plastic waste and how to avoid usage of synthetic polymers in consumer products. This is very important. So yes, the question is right. The person who has asked the question, yes, we have changed to the natural polymer now, but it is not 100%. It is to some extent only in certain area, only in certain sector, but most of the sectors still, the synthetic polymers are still being used. It's very difficult to abolish totally 100% synthetic polymer. So find a way out how to manage this plastic waste and how to manage environmental samples. So these are the answers that I would like to give for the question.
La respuesta por esta pregunta es sí hay plásticos degradables ahorita en el mundo, pero todavía no está 100% completo. Sí está utilizando plasticers todavía que puede afectar el ambiente y los organismos, todo. Hay una posibilidad de synthetic fiber o la fibra sintética que puede cambiar por polímero natural. Y cuando si hacen este cambio también no es 100% completo. Y cualquier manera nosotros hay que buscar la manera cómo puede manejar esta basura de plástico. Y en futuro puede pensar o puede buscar la manera cómo puede cambiar la forma de polímero sintético por polímero natural. Y todavía no puede decir, es totalmente está quitado este tipo de polímeros. Todavía sí existe. En algunos sectores ya están cambiando, ya sí quitaron, pero todavía sí existe. Gracias.
Bueno, vamos a la siguiente pregunta, doctor.
Yes.
Erradicar el uso del plástico, bueno, Marlene Peña. Erradicar el uso del plástico definitivamente es una posible solución para el problema. ¿Cómo afecta esta decisión en la actual forma de vida humana?
The question is how we can eradicate plastics or how it will not affect the human health.
Yes.
The question is from
Yeah,
Marlene.
eradicate is impossible. Yes, eradicating something which is already invented and already being used in use is very difficult. But we can manage plastic pollution so that it will not affect human health. So here the point is we need to know the better management system, the better waste management system of plastic, how we can manage, whether we can do a recycling of the plastic. This is one question. Or the plastic recycling in the sense we need not to change the plastic into different product. Like for example, like PET bottles, the Pepsi and Coca-Cola. Why they make Pepsi and Coca-Cola in plastic bottles? Why don't they make in bottles? Why they go for plastic bottles? So these kind of things they can avoid. The jam bottles, there are plastics. So we can avoid usage of plastic, especially when we are using this plastic in consumer products. Don't stuff consumer product with plastics. So we can avoid that. The other way around is we can try to manage the plastic. The answer lies in management. The better management of plastic waste is always very good. If we do it properly, then that is the only solution to avoid plastic from getting into human health aspects.
Thank you, doctor. I will try to tell that. Erradicación de plásticos es imposible. Cualquier manera vamos a estar utilizando y hay que buscar la manera cómo puede manejar la contaminación de plásticos. Es una muy buena opción para buscar cómo puede tener un manejo de sistema bien para manejar plásticos. Y la otra es reciclaje. Reciclar el plásticos. ¿Cómo puede reciclar? ¿En qué manera puede reciclar este tipo de plásticos? Por ejemplo, hay Pepsi o Coca-Cola que están utilizando las botellas de PET. En ese lugar ellos pueden pensar y usar las botellas de vidrio que puede evitar. El otro aspecto es, puede evitar poner las comidas o todas las cosas en plásticos. Esa es una manera para evitar usar plásticos y aparte manejo de plásticos, y también puede evitar este tipo de plásticos que no entra en la salud humana o en el sistema de humano. Muchas gracias.
Thanks. Vamos con la siguiente pregunta del doctor Jonathan. ¿Cómo controlar el uso del plástico en el futuro? ¿O cuál es la próxima elección de material para poderse usar en un entorno libre de plástico? Y bueno, si tiene alguna idea respecto a esto, profesor.
How do you control the future use of plastics what are the options which you can give to use plastics?
Future use, we should not use plastic in consumer products. No plastics in consumer products. And recycling of plastics. Recycling of plastic is very important. No single-use plastics. No single-use plastics. So these are the three important aspects. If we do, then we can reduce plastic pollution.
Thank you, doctor. La respuesta es, no puede usar los materiales de plásticos en las comidas o productos que nosotros estamos consumiendo. Ese es el primero. La segunda es, hay que buscar la manera, cómo puede reciclar ese tipo de plásticos. Y la otra es, no puede producir o no puede usar plásticos que va a usar nada más un tiempo. Hay que buscar la manera múltiple. Muchas gracias.
Ok, gracias, Jonathan. La siguiente pregunta, Juana María Castro, del CECIT 6. ¿Se han realizado estudios del efecto de los microplásticos en plantas?
Have you done experiments of microplastic in plants? It is a question from Juana María Castro. It is from
Yes.
IPN.
Yeah, we are doing that. We did a study. That's what one or two slides I have pushed on microplastic, these nanopolystyrene that we did with allium sipa, that is onion root tip. We studied to check whether it causes any genotoxic effect to the plant. But some of the studies are underway using model plants in our laboratory, because this lecture is on microplastic, nanoplastic to human and animal health. So I did not post much results on plant study.
La respuesta es, ya hicimos algunos estudios en plantas. Un ejemplo es de raíz de cebollas. Ya hicimos, yo le presenté algunos diapositivas en mi presentación y si puede revisar ahí en la presentación. También hay estudios genotoxicidad en mi laboratorio. Todavía seguimos en ese trabajo. También hay otros estudios, seguro si van a dar muy buenos resultados. Es sobre nanopoliestereno. Muchas gracias. Muchas gracias.
Bueno, pues le damos las gracias a nuestro invitado por su estupenda conferencia. We thank your guest for his wonderful conference, doctors. Y bueno, por supuesto, les agradecemos mucho a quienes nos siguieron por YouTube. Los invitamos a que a la siguiente actividad de la cátedra Héctor Mayagoitia Domínguez, con la conferencia Incursión de agua de mar en una zona costera del sur de la India. Un enfoque integrado y una solución ingenieril para la mitigación de la misma. Con el doctor Subramani, profesor del Departamento de Geología en la Universidad de Anna de la India, la cual se llevará a cabo mañana a las 10 de noviembre a las 10 de la mañana, hora del centro de México, por este mismo canal. No se la pierdan. Gracias a todo el equipo encargado de la transmisión, Héctor, Jaime y Noé. Cuídense mucho. Y les vuelvo a recordar, pueden seguirnos en nuestras redes sociales. Y por último, por favor, quédense en casa. Gracias.